So for those of you who just joined, we have a Git repository that you should have received a link to in your email. If you'd like to follow along with this tutorial, this is the June 16th tutorial on Unix pipes. Just click this Launch Binder button. And after a couple minutes, you will be taken to a Jupyter Notebook interface that looks kind of like this. So first part is a little presentation. I'm going to go over these first few slides in Reveal.js slide format. This isn't a Jupyter Lab tutorial, but if you're ever interested in turning a Jupyter Lab notebook into a presentation, there is a feature export notebook as, and then you can export it in multiple formats. And so I've picked Reveal.js for this. Oops. Okay, so this tutorial is on Unix pipes, a very useful tool in any bioinformatician's toolkit. Unix pipes have been around for a long time, since the early 1970s, and they have been widely used and one of the real fundamental tools in the Unix toolkit ever since. Oops. What this lesson will cover is specifically the use of Unix pipes in shell scripts, which probably everybody here writes either when running a sequence of several commands or when submitting a job script to a cluster like Canon, to improve the efficiency of some aspects of bioinformatics workflows. This tutorial isn't meant to teach you everything about pipes, but it's just to get you familiar with the concept if you aren't already, and to inspire you to go seek more information and find out where you might profitably use this in your workflows. And we're going to include an example on part of a short read alignment pipeline. This isn't going to specifically cover all aspects of aligning sequence reads, but it's just used to illustrate the concept of pipes and where they might come in useful. So this also will not discuss in depth all the software utilities used, but if you're interested, here's the short list of programs we're going to use. Curl for downloading files. Curl comes standard in every Mac OS system, so you've probably have it if you have a Mac. gzip, a ubiquitous data compression program. FastP, a sort of FastQ++ utility that my colleague Brian Arnold informed me of, and I think it's even written in C++ instead of Java, for pre-processing FastQ files. BWA, which is sort of an old school short read sequence alignment program by Hung Lee. And SAMtools, also originally started by Hung Lee, which is sort of a Swiss Army knife for dealing with sequence alignment data. So the motivation for learning about Unix pipes. So bioinformatics workflows, as many of you are aware, can be big, involving big data, big input files, many stages in the workflow. You might be duct taping many software tools together, sometimes by different groups. And one approach that's frequently utilized by especially beginners is to just run each stage of the workflow one after another, writing all the output to some persistent storage to disk, and then reading it in the next stage. And certainly that's a valid way of going about a bioinformatics workflow, and there are tools that automate that to some extent, like there was a SnakeMake tutorial the other week. So this is one example of half of a bioinformatics workflow. This is a variant calling pipeline that my colleague Brian Arnold put together. There's the link to the GitHub repo below. He did his using SnakeMake, but basically you start out with some a reference genome, use FastP to do some pre-processing on your FastQ files, alignment, some kind of post-processing, and at the end you have BAM files that are used for the second half of the workflow, the variant calling stage. And we're going to take this workflow, and I've translated it a bit to use Unix pipes instead of SnakeMake. However, I've also done some tool substitutions, in particular Picard. That's a Java application, and our Jupyter notebooks are limited to 2 GB of memory. JVMs can use quite a bit of memory, and it might not work to run multiple JVMs concurrently. So we're using SAM tools instead. So there isn't a whole lot of technical background that's needed for this demo, but one fundamental concept in Unix is a file. A file is really the universal interface for doing I/O operations in Unix. Unix tries to make about everything look like a file, whether it's a device driver or an actual file or sometimes memory. And it provides the same low-level system calls that allow an application to interact with a file. In particular, there are 3 special files that every process, which is essentially a running program, starts out life with. When a process is started, it has 3 open files: standard in or standard input, standard out, and standard error. It has standard in open for writing, and these definitions are directly from the POSIX standard, but it's intended to be used for primary data input, standard out for primary data output, and standard error for diagnostics. One more term that you don't hear utilized often is the concept of a filter, but that just describes an application that primarily reads its data from standard in and primarily writes it to standard out, and does some transformation on the data stream. And a lot of Unix utilities can act as filters. One common example is Grep. I'm going to switch back to my terminal here. Hold on. So Grep, to use Grep, you have to specify a pattern, but the file arguments are optional. So if I say grep foo, by default, standard in, standard out, and standard error are connected to the terminal. So if I say boo, okay, that pattern doesn't match. Foo matches, food matches, etc. And then you can hit Control D when you're done. So in that instance, grep acted as a filter. One operator we're going to use later in the presentation is the output redirection operator, this little right angle bracket or greater than symbol. That will redirect the standard out of a process to the file that's named on the right side of that operator. So in this case, if we wanted to notice that nothing was emitted to the terminal. Instead, the standard out of grep was sent to the file. Okay, so pipes. This is the main course of the presentation. So pipes are a mechanism of for inter-process communication, specifically one-way inter-process communication between 2 processes. Pipes are really the original form of inter-process communication in Unix. They've been around since the early 1970s. Very commonly used, very useful just in day-to-day command line usage. But how they work is that data is written on one side of the pipe sequentially is read on the other side of the pipe. Usually the writer writes its data to standard out and the reader reads its data from standard in. And how the coordination works, let's say that a writer wants to write a bunch of data, but a reader is not ready to read. What happens in that case? Well, the operating system kernel blocks the writer. So the system call, the underlying write system call that the writer executes blocks until the reader is ready to read some data from it. And similarly, the reader blocks until the writer has data to write. So in programming languages, in today's presentation, we're going to use the shell programming language. The pipe operator is represented by the pipe operator. But pipes can be used in almost any programming language that can trace their history to Unix, including awk. With Python, you can use subprocess.popen to execute a command and open a pipe for reading. And R also has a base, a function called pipe in base R. This is different from the pipe operator that you get when you use the magic iter package, particularly if you're using tidyverse. But the concept is kind of sort of similar, except for instead of dealing with arbitrary byte streams, you're dealing with objects in R or data internal to R. So a shell pipeline is a sequence of commands that are separated by the pipe operator. And essentially the standard output of the command on the left of the pipe flows into the standard input of the command on the right. And you can chain together pretty much an arbitrary number of commands utilizing the pipe operator. So why would you want to do this? First of all, I/O efficiency. The data flows directly between the processes without being written to disk. That can be a huge win in bioinformatics when you're dealing with files that are, gigabytes in size, and you want to, let's say, take FASTQ files and do some trimming. So maybe you start with tens of gigabytes of data and you just trim a little bit. And so you have tens minus some, 10 minus some number of bytes in the output. Instead of writing all that to disk, you can write that directly into the next program in the pipeline, maybe your sequence aligner. The other reason is concurrency. All processes in a pipeline execute concurrently. And so in particular, if you're on a multi-core machine, which since the mid-2000s, they've all been multi-core machines, those processes can execute concurrently and achieve some kind of speed up in addition to the efficiency from not writing data to disk. So an old example of this is from actually the concept of pipelining in microprocessors, which is loosely analogous to Unix pipelines. But instead of, so this would be like the naive approach of doing laundry. Let's say you have 4 loads of laundry to do. Well, you can put the load in the washer and then put that load in the dryer and then fold it and then put the next load in the washer, the dryer, and fold it and so forth. And that's sort of an inefficient way of doing laundry. Whereas if you pipeline that, put a load in the washer, after that's done, put it in the dryer, then put the next load in the washer. In stage by stage C, you're at the point where you have you're folding one load of laundry, you have one in the dryer and one in the washer. And Unix pipelines are very similar in concept. Okay, so now we get to the point where we go back to the Jupyter Notebook. Scroll down a little ways. Okay, so these are the concrete examples that you might actually find useful. So here we're going to do a sequence alignment pipeline using relatively small input data, but the first example is we're going to fetch data, uncompressed FASTA file using curl, compress it on the fly using gzip, and write that to a file. We're naming it something reasonable, essentially the input file with a .gz suffix, but we could call it anything. So curl by default writes its data to standard output, which we're using this pipe operator here to pipe to standard input of gzip. If you don't give gzip any file arguments, it reads presumably uncompressed data from standard in and writes it to standard out. We're specifying a couple options here. -v verbose just indicates the compression ratio at the end of gzip execution. It will emit that to standard error. The -9 is for maximal compression. This isn't commonly used, but if you have some reference data set that you're going to utilize over and over, it makes sense to compress that maximally, especially if you're like submitting sequence to some kind of repository or for publication. It makes sense to compress that maximally. You're spending some time beforehand, some extra processor time compressing the file, but you know, then ultimately it'll take up less space on disk, and every time somebody downloads it, it'll use less network bandwidth. So, sorry, a little bit of a tangent. So to execute, oops, so I have no kernel here. If you see this no kernel message, click on that button and select the bash kernel. So all of these cells will be executed by bash. And then the way you can execute a cell is either by pressing the play button here, or Shift Enter is a handy keyboard shortcut. Okay, so here we have, this is the output of gzip. It just says that we compressed this file, made it 71% smaller. So it's 29% the original size. And then this just indicates that this is the output of the ls command showing that we wrote this file. So, okay, so that's our input. It's been written to this directory here called input. So now that we have this input FASTA sequence, let's find out the sequence IDs and the length of every sequence in that file. And we can use a utility called awk for doing that, a standard Unix utility. Danielle had an awk tutorial a few weeks ago, and that is also in this Git repo. But awk can't read compressed input. Awk only knows how to read text. What we can do is decompress, but awk does know how to read from standard input. It's commonly used as a filter. So what we can do is decompress our compressed FASTA file using gzip with the -dc option, D for decompress, and C to write to standard out instead of creating a new file without the .gz suffix. And then we pipe that to awk standard in. And you don't have to understand what all of this is doing, and it's actually a little bit more complicated than it needs to be. But basically, this accepts a potentially multi-FASTA file as input, where each FASTA sequence can be represented on multiple lines. And it determines the sequence length for each and writes that to standard output. This FASTA file is a bit simpler in that the entire sequence appears on one line. So this awk program is a little more ornate than it needs to be, but it's still a useful program to have. I actually have a shell function in my .profile so I can execute this. Okay, so there are 3 sequences in the file, sequence ID 1, sequence ID 2, sequence ID 3, and they're all of length 200,000 bases. Okay, so now that we have our reference genome, we need some sequence to align against it. So we're going to use the curl command for that again. This just illustrates that curl, we're going to fetch 2 FASTQ files. This little pattern just indicates that curl can, with a single argument, curl can be used to fetch multiple files. Here we had an _1.fastq and an _2.fastq. Rather than writing 2 curl commands, I just fetched that as part of the same command. And if you have a lot of files, that can be a handy little shortcut. Okay, so we have our fastq commands. Next, in order to align sequence against it, we have to index our reference genome. And there's a command called bwa index to do that. BWA stands for Burroughs-Wheeler aligner, and it computes something called the Burroughs-Wheeler transform. That's what this BWT stands for. And that was done really quickly. So yeah, it constructs a suffix array from the Burroughs-Wheeler transform. Okay, so now we're ready to go. Okay, so this is our naive or sequential sequence alignment workflow. Just to go over this really quickly, we trim the reads, the FASTQ reads, we align them using BWA mem. We sort the reads by alignment by read name, or sort the alignments by read name, excuse me. That's needed for the SAMTools fixMate command to fill in the mate coordinates in the alignment records and the insert size. Then we have to sort it by coordinate because that's required as the input of SAM-tools mark dupe, which marks the duplicate alignments in the flag field. And then we indexed the resulting BAM file for further use. Okay, so let's run this. Okay, now we're on BWA-mem. Yeah, and this set plus O xtrace is just kind of a handy option to print every command to standard error before executing it. Really handy to include in job scripts if you want to see what commands are executing. Okay, so that is done. It took about 27.5 seconds to execute. That was what this time command did. So this time is a shell built in, and we use this special shell syntax here to run all of these commands in a subshell, which kind of, it's sort of like stuffing all of these commands into a shell script and executing it. It runs all of these commands in a child process. But this time command times everything that runs in this subshell. And so that was the resulting time. Okay, so let's see what the damage is as far as disk space. Okay, so we can see that there are 90 megabytes of files on disk. If we were going to be meticulous, we could actually remove these intermediate files that we don't need from the previous stages. But in practice, unless you're using an automated pipeline like SnakeMake that automatically does that, I rarely see that in practice. Okay, so before we do the optimized pipeline version, I'll just mention that not all utilities can read from standard input. BWA-mem is one example. That is, it can't explicitly read from standard input if you leave one of the file arguments off. So if you type BWA-mem without any arguments, it prints the usage. And how you parse this is BWA-mem accepts some options that are optional by definition. It has a required operand or argument, the prefix of the reference sequence that you're aligning against, prefix being our fasta.gz file, and presumably there are some other files with a suffix that were created by BWA index. And then our reads. And notice that this reads argument is not optional, it's required. Some Unix utilities can accept a hyphen in place of either of some required file argument. But BWA-mem, that's not a standard, that's just a convention. And BWA-mem in particular doesn't understand that. So we can use /dev/standardin to mean the special file associated with the BWA process, to essentially mean BWA standard in. So if we see BWA-mem, the database prefix /dev/standardin, BWA will read its reads from standard input. And special note, this only works if the utility can read its input sequentially. Sequence alignment is a sequential process, so that's not an issue, but not all utilities can read data from standard input, can read input sequentially. Like for instance, if you're using SQLite 3 to interact with a database file, it's going to need to seek to different parts of that database file. But a lot of what bioinformatics involves is reading data sequentially. Okay, so here's our workflow. I'll just go over this quickly. So this uses one big pipeline, essentially. The process in each stage in one stage, writes its data to standard out. That's read in as the input to the process of the next stage. And all processes execute concurrently. I've annotated it with comments. Just to go over it briefly. So we're using a special mode of FastP. Instead of writing the trimmed sequences to separate files, so these are read pairs, paired end reads. If you specify the -- standard out option, FastQ, or excuse me, FastP writes them in interleaved mode. So you'll have read one, read two, read one, read two, all in a single data stream. BWA mem, when you give it only one file argument, it assumes that, or it can detect that these reads are interleaved by the read names. So BWA mem writes its output to standard out by default. SAMTools sort, if you don't specify any file arguments, it acts as a filter. It reads its data from standard in and writes it to standard out. By default, SAMTools sort uses the same output format as it read from standard in. We don't want, so it'll read SAM, which is a text format from BWA mem. We want to use BAM for further processing. BAM is a bit more efficient. It's converted into a binary format that essentially SAMTools uses internally in memory already, and it doesn't have to spend a lot of time processing text in subsequent stages, because text can be expensive to process. So what we're going to do is convert it to BAM at this point, but also specifying using compressed level 0, we're going to use uncompressed BAM, so that SAMTools doesn't have to spend CPU time uncompressing the BAM. It can immediately read it in and essentially represent it in memory. So SAMTools fixmate also acts as a filter. SAMTools fixmate does understand this conventional hyphen argument. So we don't have to use dev standard in and dev standard out here, but we could have, we wanted to. SAMTools sort, again, reads from standard in, writes to standard out. And if we don't specify the -n option to do name-based sorting, it'll sort in coordinate order. Then we're using markdup to mark duplicates, writing some statistics to an output file. And then we're using maximal compression from here on out, because this is going to be our resulting BAM file that presumably we're going to use multiple times. Excuse me. And so this last part, we're introducing T and process substitution. So I'll go over that in a bit, but first let's run this. Okay, so notice that all of these are running concurrently. I'll illustrate this again here in a second. Okay, and we're done. So it took 17 seconds instead of 26.5 seconds. And if we look at the amount of data, this is the sum total of data that was written to disk, 11 megabytes. Essentially, most of that was our output, in addition to this FastP HTML file report and JSON file. So to illustrate this one more time, I'm going to run this. I'm going to use the top command so you can see this execute. -C to print the arguments for each process. And then I'll hit uppercase V here to show the process hierarchy. Okay, so I'm running this again. Okay, so here we can see all of these processes running concurrently. So now BWA mem is running. You can see the CPU utilization. Okay, so, now we have SAMTools sort and fixmate. Sort is writing output to markdup, it looks like. And yeah, now we're done. Took a little bit longer that time, but essentially the same amount of time. Okay, So what was the special syntax that we were using here? This gets off into the weeds a little bit, and you rarely see this in informatics workflows, but I'll introduce it just so you're aware of the concept, and you might find a use for it at some point in the future. So instead of, so T basically takes its standard input and writes it to standard out, kind of like the cat utility. But in addition, it duplicates that and writes it to one or more files. In this particular case, what we're doing here, so this is the last stage of the pipeline. So we have samtools markdup, which reads uncompressed BAM, and we're writing maximally compressed BAM to T. So T is reading this compressed BAM from standard in, writing it to standard out, or writing it to a BAM file, first of all, out.bam, and this is our final BAM file. Instead of just writing this to output and then reading it all in later and for samtools indexing, because samtools can't index, samtools markdup can't write an indexed file. It can't generate the index as part of the samtools markdup command. So one option, not the naive option, but I guess the common option would just be to write the output BAM file, then read that all in again, you know, tens, possibly hundreds of gigabytes, and index that. And then run samtools stats on that in a separate process, read all of that in and write it out again. But we're sort of making a clever use of T here to write our final output BAM file. The standard out of T is going to samtools stats to generate the alignment.stats file. Or I guess we're calling it out.bam.stats. And we're also using this special funny syntax here. This is called process substitution, but this sort of creates another pipe to this process, samtools index, which is reading from its standard input and writing its output to this index file. This is a little bit more of an involved concept that I have here in the bonus material, and I can explain further if you'd like, but I just thought I would demonstrate that here just for your enjoyment. Okay. So how are we doing on time? Okay, so a little long, but okay. So are there any questions thus far? Would anybody like me to explain this concept of process substitution a little bit further, or anything interesting about the pipeline that you would like me to go over, or anything that you're currently working on that you would like some advice on? I'm open for questions. Oh, and Tim, are you going to stop the recording at this point?
Yeah, just need a.
Okay.